This is TWIS, This Week in Science, episode number 535, recorded on Wednesday, October 7th, 2015. Celebrate the octopus! Hey everybody, I'm Dr. Kiki, and this is This Week in Science. This week we are going to fill your head with prizes, ruses, and octopuses. But first...
Disclaimer, disclaimer, disclaimer. It has happened again. Another campus, another mass shooting, another group of families grieving, while we suffer another round of political chest pounding and promises to do something, or vows to do nothing. That it so often takes place while people are seeking education, performing the act of learning, an act that propagates knowledge from the past into the future, the act of learning that preserves our understanding of the world and maintains all of our collective history. Education is a birthright. Without it, we lose our science, our medicine, our technology, our history, and our humanity. It is the one right we share that truly keeps us safe and should be defended above all others. And if there is any more sacred place on this planet than a classroom, I have not seen it. Hopefully, we won't end this latest lesson with yet another moment of silence. Too much is at stake to make that mistake again. This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to Kirsten and Blair.
Good science to you, Justin, Blair, and our guest, Dr. Richard Ross, and everyone out there. Welcome to another episode of This Week in Science. We are back yet again to spread the knowledge. Right, Justin?
Yeah.
Doing it. Yeah. So everyone, I hope you're ready to hold on to your pants, your hats, I don't know, all the things that get blown away when you get excited by science.
Your tentacles?
Your tentacles.
Hold on to your tentacles.
Yeah, because did you know that tomorrow, October 8th, is World Octopus Day?
No, I get it. October 8th. Got it. October.
But October's the 10th month. Shouldn't it be August? No, I guess that makes in the literary as well as in numerical.
It makes enough sense. And it kicks off Cephalopod Week. And so in honor of World Octopus Day and Cephalopod Week, we have brought a cephalopod expert, Dr. Richard Ross, to the show tonight, and he's going to be joining us to talk about octopuses, their behaviors, and the mating habits of the Pacific striped octopus, which is a very special octopus. So thank you so much for joining us. We'll be talking to Richard in the second half of the show. Everybody who's out there. Yeah. In terms of science news, I brought psychic robots, sex, and Nobel Prizes. That's what I've got. Justin, what did you bring?
I've got old hands, old feet, and new eyes.
Interesting. And Blair, what's in the animal corner?
I brought fecal fakery, nutritious nuptials, see if I can do this, and nasty nuclear fallout?
Very nice. All this alliteration and more here on TWIS. But let's jump into it. Who won the Nobel Prize?
Was it me? I forgot to check the mail. I should go.
Yeah, you didn't get the phone call. If you won, you would have gotten a phone call very early in the morning, and you did not. So, nope, not you. But there were some amazing scientists who have done amazing, groundbreaking work, and who have been showing us more and more about the world, universe that we live in. The Nobel Prize in Chemistry was given this week to Thomas Lindahl, Paul Modric, and Aziz Sankar for their studies of DNA repair. And the interesting stuff that they've been working on are different mechanisms that fix DNA when it goes wrong. So there are various ways that DNA gets messed up. Mutations occur when DNA is replicated, when it's copied. Other times UV rays, sunlight, cigarette smoke can actually cause mutations in the DNA. And when I say a mutation, what that means is that DNA has what are called nucleotides. And the nucleotides line up kind of like a zipper. They fit together and they match. They have another nucleotide that they just fit like puzzle pieces with. But sometimes if a UV ray comes in and strikes just the right nucleotide, it'll mess up that connection. And it's kind of like getting that weird little tooth in the zipper that goes wrong, and your zipper never zips quite right again. So these researchers have been discovering how DNA, or how our cells, go about fixing themselves when these problems arise. One of the aspects of this is being able to come up with new methods for treating cancer, maybe even one day curing cancer. Other aspects have to do with cancers that are an issue from birth, that they're genetically oriented as opposed to something that occurs through experience in the environment. So these researchers have made some amazing advances in this field, and so we congratulate them very much. And in physics, the Nobel Prize for Physics was given to, drumroll, Takaaki Kajita from the Super Kamiokande collaboration, and Arthur B. McDonald from the Sudbury Neutrino Observatory collaboration. And these researchers discovered that neutrinos oscillate. And what does that mean? Does it mean that they shake, rattle, and roll? No, it means that they change flavors. They change from one kind of neutrino into another. And so part of this discovery helped explain, why all of our ideas about how many neutrinos should be passing through the Earth and through our detectors, and what kinds of neutrinos we should be detecting, why we were getting the wrong answers. Basically, the wrong kind. We were coming up short in our calculations of muon neutrinos. And the reason is that they change and they turn into different kinds of neutrinos. And what's important about this is that means that neutrinos have mass, and that this goes on to tell us all sorts of things about matter in the universe. So this discovery of the shifting nature of neutrinos tells us much more than we bargained for. It tells us so much about how our universe is put together, really. And then the Nobel Prize for Physiology or Medicine has to do with parasitic infections, and it was shared by William C. Campbell and Satoshi Omura for their discoveries related to parasites that are caused by roundworms, not parasites, diseases caused by roundworm parasites. And additionally, UU2, the only female in the Nobel Prize Science Awards, for her discoveries about Artemis. I can never say the name of this drug. Artemisinin anti-malarial drugs. Her discoveries about that began with a plant and ended up resulting in a drug that can be used to combat malaria. Have given us all of these scientists working on these different drugs and different treatments for parasitic infections are really helping our global population fight back against the diseases that ravage us, especially in the more impoverished areas of the world. Yeah, so these are the wonderful, wonderful things that have received awards, wonderful scientists, people who have received these awards. And if you want to read more about all of this, you can go to nobelprize.org. nobelprize.org. And you can read all about it. And they've got some great sound clips from previous winners about how they felt upon receiving the award, and it just kind of gets at that human heartstring angle of these people are really excited to get to be honored in this way. Oh, yeah. It's pretty cool.
I'll have to wait another year.
Yeah. Yeah. Oh, and additionally, as Strengths is bringing up in the chat room, We can't forget the Ig Nobel Prizes, especially the Ig Nobel Award for discovering that is universal to all languages.
Is it really? Oh, that's
I'm
fascinating.
not surprised.
All languages have some form of it must be very important. That what? I don't get it, It's important.
That actually might be
world round.
Might actually be the first word if you think about it.
Could be.
Because somebody else came up with the sound, and maybe they were trying to communicate, but you're not sure. And so the first word likely that caught on was huh?
No, wouldn't you say the first word first, and then you didn't hear it right, and then you would say huh?
No, because the first one that everybody agrees on is, because everybody's got to invent first words, but if you're the first one to invent that first word, it's really not a word yet. It's just a sound you're making that you have a meaning for that nobody else does. The one that we all seem to have agreed upon is huh?
Huh? Exactly. Now, moving on into other news, I have a story about the giraffe. Now, this is one of those things that makes you go, huh? Everybody wants to know how the giraffe got its long neck. Mm-hmm. Where did that come from? Stretching and reaching to get to the trees.
Yeah, that would be Lamarckian.
Well, researchers did a genetic analysis and have looked at fossil cervical vertebrae from existing and two existing species, one of giraffe and one related to giraffe, and some extinct species as well. They studied 71 fossils of 9 extinct and 2 living species in this family of giraffes to kind of figure out what is going on with the giraffe neck. Where did it come from? And it turns out that the giraffe neck did not just elongate all in one go. It wasn't a jump from short neck to long neck, but it happened in stages. And it started out with the C3 vertebrae elongating early on, around 7 million years ago. The cranial end was what started the stretching 7 million years ago in a species known as Samotherium. And then there was a second stage of elongation that occurred in the more tail, the caudal portion, the portion of the vertebrae closer to the tail, about 1 million years ago. And so it's these different stages of elongation of these vertebral bones that actually led to the modern giraffe's lengthy neck. Now, another member of the giraffe family, the okapi, is the only other living member of the giraffe family, but it didn't evolve a long neck. And so the researchers say, it's one of 4 with a secondarily shortened neck. So it has a completely different evolutionary pathway, which is interesting. Why did the okapi end up going down this different pathway after, ancestors had started elongating the neck? Why did they shorten it, and what evolutionary pressures would have led that to occur? Interesting questions.
And I'll stick with the Lamarckian on this one, just for one.
Just because you always want to stick with the Lamarckian.
The internet froze him because he was wrong. So, no, what I love about giraffe necks is that I learned. You did. I learned in college that just in the past 20, 30 years, we found out that the giraffes, everyone assumed that they got their longer neck so they could reach the tippy top of the trees. But in doing behavioral studies on giraffes in the wild, they found that consistently giraffes did not eat from the tallest leaves they could reach. They were eating from what was right at the middle of these trees. And they found that actually the long, thick necks were being used in battle with each other, the males.
Right, and which we've seen where they swing their heads on the ends of their neck and they bash each other in the neck.
Right, so, and that the length was actually a predictor for who was going to win in these fights, and not actually a predictor in which leaves they would try and eat.
But you're looking at the wrong end of the giraffe. Couldn't you say that the reason for the long legs is so that the neck was just closer?
And so, Justin, the next question that these researchers are going to look at are the long legs of the giraffe.
There we go.
Which came first, the neck or the legs?
Yeah, so we will actually discover the timing if they do a similar analysis of fossil, extinct fossilized bones and modern bones. We will get an idea of the timing of the elongation of the legs versus
it may have just been
in comparison. It could have just been. Look at
having
my long
a giraffe
leg.
with
My long
short
legs
and a really long neck, that might just have been awkward.
Yeah.
All
right,
That's such
Justin.
a great clue, though, the okapi. I love that. The secondarily shortened neck.
Yeah. Very interesting. All right, Justin, tell me some news.
Okay, so two papers have been published on the recent hominid fossil find of Homo naledi, appropriately titled "The Foot of Homo naledi" and "The Hand of Homo naledi." I'm going to say naledi or naledi differently every time I say it, by the way. Not sure if there is a really right way to say it yet. Taken together, the findings indicate that Homo naledi, may have been uniquely adapted for both tree climbing and walking as dominant forms of movement, while also being capable of doing precise manual fingery manipulation of things. They find the foot shares many features with a modern human foot, indicating it is well adapted for standing and walking on two feet. However, the authors note it differs in having more curved toe bones, and their feet were generally flatter than seen in the average modern human. So despite the close similarity in the foot structure, Naledi likely did not walk like us. It had its own strut that we have, I think, yet not recreated and figured out, but it's going to have a different strut than the humans. The hand in the other paper, the hand reveals a unique combination of anatomy that has not been found in other fossil humans before. The wrist bones and thumbs show anatomical features that are shared with Neanderthals and humans and suggest powerful grasping and the ability, if they so chose, I suppose, the ability to use stone tools. On the other hand, or rather at the ends of both of the hands of the Naledi, the finger bones are more curved than most early fossil human species, such as 3.2 million-year-old Lucy's species, Australopithecus afarensis, suggesting that Homo naledi still used their hands for climbing in the trees. This mix of human-like features in combination with more primitive features demonstrates that Naledi was both specialized for complex tool use activities, but still used for climbing locomotion. This is also confirmed by looking slightly outside of this study on the hands and looking at the shoulder joint that looks more ape-like than human. So despite having a tremendous number of fossils to work with, the Naledi bones found in a cave and cannot be tied to a specific geologic time. Also, because of the ancient hominid fossils that we do have good dates, because they were found in places that they were well preserved, were actually much less well preserved, and so we don't have complete fossil records of those in which we have dated. The Australopithecus afarensis foot that we know of, we believe is very human foot, but we have just a segment of bones to work off of to make the extrapolation, whereas in this case, we've got the whole bones. Because Naledi remains undateable, much like I was in high school, there is a great deal of informed speculation going on at anthropology parties about what this creature, where this creature fits within our evolutionary past, or if it is even our evolutionary past. It is possible that Naledi is older than Lucy, much older even, giving greater affinity for climbing trees than we see in Lucy. Or this could be a separate branch.
Yeah, is that the idea? Is that it's a separate branch, something that that branched off earlier than Lucy?
So it could be that. It could be a separate branch of hominid evolution, even one more recent than Lucy that simply dead-ended before we saw evidence of it. Or Lucy may be the dead-ender and Naledi is our actual ancestor. Or they were contemporaries who mingled like Homo sapiens and Neanderthal, or none of the above, and a greater mystery still awaits. That's the thing we don't know. What we do know is we have a truly unique opportunity to view with a great deal of missing link type hominid evolution steps. So this is we're getting to see how stages of evolution take place from tree climbing to walking, even though we can't place it within a linear graph of our own evolution. this is very likely a similar thing that we would see for any hominid that made this step. But yeah, there is something intriguing in the fact that the hands are so well adapted for climbing. We have better hand, I believe, of Australopithecus afarensis than we have of its feet. And those, I believe, are less tree climbing.
Yeah.
So this would sort of, if we were going to say there's a linear path from tree to bipedal tool use, this would, in that sort of scenario, put Naledi as earlier than Lucy. But this, again, it could be contemporary.
We have no idea until they can date the fossils. We have no idea.
Even when they date them, it's going to be difficult. That's the thing. What we really need is just more afarensis. We need more of
others,
more detail of the others, so we can have a hand by a hand in great detail and see the similarities and differences of thumb and pinky finger and that sort of thing, or the foot. I guess the foot's got more of the missing model there than the hand. But
yeah,
still really fascinating. And this is just the first two papers.
I know there's so much more to come.
Yes, so this is going to be an exciting story to be tracking throughout the next year.
So tell me about your next story, which is also exciting.
Yeah, so this is regarding age-related macular degeneration, or AMRD. It says it could be treated by transplanting photoreceptors produced by directed differentiation of stem cells, and that's according to Professor Gilbert Bernier, University of Montreal, and it says here, ARMD, the age-related macular degeneration, is the greatest cause of blindness among people over the age of 50. Affects millions of people worldwide, and as we age, it is more difficult to avoid. Amongst people over 80, the accelerated aging of the retina affects nearly 1 in 4. People with ARMD gradually lose their perception of colors and details to the point where they can no longer read, write, watch television, or even recognize a face.
Yeah, so in the macula of the retina, like you have the rods and the cones, your photoreceptors, the cones especially, just light receptors. So when those disappear, our bodies don't make any more. That's it. You're done. They're done. So if we could find a way to implant fresh cones and or rods into the eye, it could go a long way for helping people.
There is a way of doing that, actually. Unfortunately, though, we don't have a way of producing cones in a great level. Bernier's team, it says here, has developed a highly effective in vitro technique for producing light-sensitive retina cells from human embryonic stem cells. Oh, wait, this is what we were missing. Our method has the capacity to differentiate 80% of the stem cells into pure cones, says Professor Gilbert. Within 45 days, the cones that we allowed to grow towards confluence spontaneously formed organized retinal tissue that was 150 microns thick. This has never been achieved before. Okay, so tell me more. I guess I should tell you more since I.
Yes, tell us more.
In order to verify the technique, Bernier injected clusters of retinal cells into the eyes of healthy mice. The transplanted photoreceptors migrated naturally within the retina of their host. Cone transplant represents a therapeutic solution for retinal pathologies caused by degeneration of photoreceptor cells, Bernier explained. To date, it's been difficult to obtain great quantities of human cones. His discovery offers a way to overcome this problem. So, into the future, you can keep your eyesight, and perhaps, Blair, you have your hand raised? Yes. Maybe.
I can have cones?
Maybe.
Oh my gosh.
Maybe you can see the full spectrum of colors. Maybe one day.
At some point, maybe.
I wonder if my brain could handle it.
Oh, yeah. I wonder if your brain would be able to handle it.
Yeah, just imagine a color you've never seen before. And
I'd probably have a headache for several days, and then I would figure it out.
Or you'd be like,
so many colors.
Wait, what's the color you can't see? I forget. Is it red?
All of them.
All of them?
It's complete. The world is not gray to you.
No, it's just muted. All colors are dull.
Right.
Life
Maybe
is
you won't like it all flashy and bright. Maybe you'll be like, this is so gaudy.
Well,
this color-filled
who can tell?
Ugh.
Maybe someday I'll get some cones and I'll know what the Earth looks like to most people.
but in the meantime, I know what you do have. An animal corner!
Yes.
She loves our creature, cry that stuff. Biped, millipede, no pet at all. Wanna hear about animals? She's your girl. Except for giant pandas and squirrels. Animal corner.
I want to talk to you guys about poop today.
Woohoo!
I'm really glad that you decided to say that. Okay.
I love
Let's go.
talking about poop. Poop is fascinating, but it is especially fascinating when it comes to dung beetles.
I love dung beetles.
Yes. Now, for perhaps the first ever time, researchers from the University of Cape Town and the University of KwaZulu-Natal in South Africa have found an example of what they call fecal mimicry.
Somebody's copying somebody's poop.
So, a grassy plant known as Ceratocarium argenteum. They were looking at this plant, this grassy plant, and trying to figure out how their seeds were getting dispersed. The original consensus was that it was probably mice. Yeah, sounds good. Mice are moving the seeds. But to find out, they dropped 195 of these seeds near monitoring stations in a nature reserve and recorded what happened with video cameras. I've kind of given it away here, but they were not expecting this. Dung beetles were coming up, taking these seeds, moving them through the area, rolled them to nearby locations, buried them, and then disappeared. Now, normally, dung beetles, they grab animal droppings, they roll them to a good spot, they bury them, then they eat some of it, lay their eggs nearby, and then that's a location for food when the eggs hatch. So, what appears to be happening is that these seeds are tricking the dung beetles into thinking they're poop. The dung beetles roll them over, bury them, and then, just when they're about to lay eggs, discover the ruse. And then they give up and they leave. So, this is what gets so crazy. Upon inspection, okay, these seeds, they look a lot like bontebok, which is a type of antelope, dung. They look a lot like this dung. And, this is where it gets especially interesting, the chemical composition of the seeds resemble the dung.
Wow.
Oh, that is fascinating.
Yeah.
So there's some
yeah.
It goes just beyond physical mimicry into
Right.
like physical and chemical.
Mm-hmm. Which, if you look at the graphic that Kiki has up, you can see the top left. If you guys are watching this, if you're not watching, if you're just listening, follow the link on TWIS.org and look at the picture because it's pretty interesting. So the top left two figures A and B are the seed. Figure G is the dung. So similar size, similar hue, but the surface looks very different. And I'm thinking if I'm a dung beetle that's right up against that piece of, quote, dung, I would think it would look pretty different, which indicates to me something that this article didn't even mention, which is that would indicate that dung beetles do most of their dung picking based on chemical composition.
That would make sense. I think that would make sense.
Yeah. So that's very interesting. And then, of course, you wonder what happens to the dung beetle after they've exerted all this energy rolling fake poop around. How long does it take them? Yeah, how long does it take them to then find the correct dung and then lay their eggs? So it's a question of, is this harmful to the dung beetle's ability to reproduce, or is it just kind of like, oh, shucks, and they go and they grab another piece of dung. That's not a big deal.
I would imagine. I mean, it's fooling them, and it will reduce their ability to produce viable offspring.
And I would say the opposite. I would say the opposite. I say that these dung beetles now have gotten practice. Right? They've gotten to practice a little bit more than the others, and so when it came to the game time, they were actually better at bearing real stuff.
Right, they're pumping iron at the gym by rolling around the seeds.
Practice.
Yeah.
Oh my goodness.
Yeah. So fecal mimicry.
Well, take us into crickets.
Oh, let's talk about nuptial gifts. I'm sure we've all received gifts from a loved one in the past. Well, if you were a cricket, you would give an edible nuptial gift that was attached to your sperm packet. If you were a male, giving that to your female. The sperm packet is called ampulla, and they hand it to the female. And the female could just gobble down that sperm, and that kind of ruins the whole point. So instead, giving them this nuptial gift gives them something else to satiate themselves while the nuptial gift does its job of pumping sperm into the reproductive tract. Now, with decorated crickets, Grillodes sigillatus, they have a very interesting situation where this nuptial gift may have an added benefit. So, the University of Exeter found that this love gift is more than just nutrition to satiate the female instead of her eating the sperm, and it's also more than just a time delay. It offers both of those things, but it looks like the proteins in the nuptial gift determine molecular structure and function inside the cricket. So some of the proteins were protected from being broken down by enzymes, and they bear a striking resemblance to growth factor proteins existing in other insects, which suggests that these proteins could promote cell growth and development in target tissues within the female's body, which means they could influence female reproductive behavior.
Right. That this gift could actually be much more. It's more of like a Trojan horse.
Right. Yes. And it's possible, what they're suggesting is that this nuptial gift has something in it that causes the female to be less likely to mate with other males.
Oh, interesting.
So crickets are known female crickets are known to mate with multiple males. They have multiple suitors' sperm inside their reproductive tract. And then there's actually, in some crickets, there have been studies where it appears that the female can actually select which sperm gets used inside the female. So for all those reasons, as a male in the cricket world, it would be beneficial to reduce the number of crickets she's mating with, because then that's less competition inside. You have to worry less about living up to the other suitors' specimens. So. Right. So by giving them this special nuptial gift, not only is it keeping her from eating the sperm, not only is it keeping her from mating with other males just in sheer time alone, but it could also be changing her behavior completely.
Fascinating. Now your last story for the Animal Corner I find absolutely interesting, and I think it has far-reaching implications for humanity and how we interact with the Earth.
Yes. So let's dial our Wayback Machine back to 1986.
I remember 1986. I was there.
I was born that year, but
I know. That's why I was making the point. I know.
At this place called Chernobyl, something happened. What happened?
There was a nuclear meltdown.
That's right. And so it's been a place
nuclear vessel
that people have not gone so much since then. And so a recent study at the 1,621 square mile Chernobyl exclusion zone previous studies showed evidence of radiation effects and significantly reduced populations of wildlife. And just now in a recent study, for the first time since the accident, researchers have long-term consensus data that reveal thriving wildlife populations.
Yeah, and so the key here is that we're talking about populations and we're not talking about individuals.
Right. So it's possible that that individual moose is not feeling so hot because of the effects of radiation, but that the species is there. So they found moose, roe deer, wild boar, wolves, all of these things that you can't really find in a lot of the U.S. And this is what I think is so funny. The quote from a professor of environmental science at the University of Portsmouth in the UK. He says, this doesn't mean radiation is good for wildlife, just that the effects of human habitation, including hunting, farming, and forestry, are a lot worse.
Wow.
Yeah, it's mainly that people have not been in the exclusion zone that has made the difference.
Yeah, so doesn't matter if it's contaminated with nuclear fallout issues. It's better than being where there's humans. Yeah.
So if we had a chance to retitle the show, I might retitle it "Things That Make You Go,
Humans.
Yeah.
Worse than nuclear fallout.
Yep, that's basically what the moral of this story is. Which, it made me go real hard.
Yeah.
Which is why I put it in the rundown. It's so interesting to take a step back, think, okay, the effects of human civilization are more devastating than a nuclear meltdown.
And on that note, we're going to take a break. This is This Week in Science. Stay tuned. We have our interview with Dr. Richard Ross coming up in just a couple of moments. Stay tuned, please. Hey everyone, you want to help out TWIS? Buy our stuff! That's right, TWIS has merchandise that you can find in our Zazzle store. So if you head over to twis.org, click on the Zazzle store link that's in the main header bar, and you'll find hats and t-shirts and tote bags galore, lots of things, even Christmas ornaments. I can't believe I just said that. It is October, Christmas is coming, the goose is getting fat. Order some of our stuff and support us in the process. And additionally, we are supported by donations from listeners just like you. Your donations pay for everything that we do. New light bulbs, our hosting bandwidth that we need to get this show on the road, everything that we do, really it's you who keep this show afloat. Any amount that you're able to give, $2, $5, whatever, it really helps. And so you can make donations a couple of different ways. We have PayPal buttons all over the website. So go to twis.org, PayPal buttons, bloop, 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 donate, happy, done. We also have a Patreon account. So if you go to Patreon, patreon.com/thisweekinscience, you'll be able to support us per episode on an ongoing basis at whatever level you choose. And sometimes you might even get little gifts in return. Whatever your preference, help us out. Go to the website, click on these buttons, PayPal, Patreon, and support us. If you can't afford a donation, can't afford to buy stuff, we totally understand that as well. We can always use your help, though. So anything that you're able to do to help us out can really benefit what we do. Help us get a bigger audience, which can then get us more donations by telling your friends about us. Tell your friends, families, share science with the people you love. We thank you for your support. We really could not do this without you. And we're back. There's more this week in science for you. And now we are going to talk about octopuses. I'm so excited. Are you excited? I'm excited. I would like to welcome to the show Dr. Richard Ross. He's a senior biologist at the Steinhardt Aquarium in the California Academy of Sciences. He's a cephalopod expert and has authored many academic papers, most recently on the Pacific striped octopus, which we are going to discuss a bit today. He not only takes care of animals at the aquarium, but also has massive tanks of his own at home. And he's worked as a juggler and a corporate script writer. I also see that he's done improv. So I'm thinking this is the kind of multi-talented person that we like to talk with here on TWIS. Thank you so much for joining us.
Thanks for having me on and saying nice things about me.
You're welcome. You're absolutely welcome. So tell us about your role in working at the aquarium at the California Academy of Sciences.
I call myself I call what our group does, we're husbandry scientists. So our job is to keep stuff alive and keep it happy and breeding. And it's actually interesting when we're in the field with what I guess you could call traditional researchers, and they're going to pickle something, and we say, no, give that to us, and we'll keep it alive. And then when it dies on its own, then you can have it and pickle it. But let us keep it alive. And I think that's exciting stuff, and I think it's important as we move into, where science all seems to be going about sustainability. Well, it's good to know the taxonomy of things, but it's also good to keep them alive and breathing. So I feel pretty good about the work we're doing.
Yeah, it is interesting to think about the different kinds of science and the fact that historically, scientists, biologists have gone out and just any animal we want to study, we kill it.
Right. And then at the end of our study, we kill it. So we're working on changing that slightly. So.
Awesome. So the octopus, various different species, are they really hard to keep alive in captivity and to get them breeding, behaving normally?
That's a two-part question. It doesn't have to be, but I'm going to complicate everything.
Perfect.
If you can keep a reef tank, if you can keep corals and things like that alive, octopus isn't that much harder. But that first part's not that easy. So I guess the short answer is, yeah, they can be pretty difficult. There's a lot of stuff you got to keep moving correctly all the time. Mm-hmm. And then you've got to feed them, which is difficult because they often will only eat live foods. So that can be a big pain. And then keeping them breathing is a whole other thing. And then breeding them is a whole other game because some of them are just you can't do yet.
Yeah, well, I know I've worked with birds, and in different species, say, of the corvids, scrub jay versus a raven versus a crow versus a magpie, some, and even different closer relatives of the scrub jay, some do really great in captivity and others don't. Like, they're all fairly closely related, but some of them just don't take to it. In fact, Steller's jays will just smash their heads into the cages, and they'll pretty much kill themselves if you put them in if you get them in a captive situation. Do you see stuff like this happening with with the different octopus species?
To an extent, but it's more of giving them what they need. I did a talk once and talking about how hard it was, and we couldn't get these baby octopuses to eat and survive because that's really hard. And a lady in the audience said, "Why don't you just give them what they need?" And it made us all laugh hysterically. Because we don't know what they need. We're trying. That's kind of our goal. So they do different things. I don't know if your corvid analogy works as well with that. I think the biggest thing that happens is people think octopuses are all the same. Mm-hmm. So they try to keep them the same, and that just won't work. So with the Steller's jay, clearly you need a huge enclosure to minimize the head bashing.
Exactly.
So with a bigger octopus, you need a bigger tank, and they're all different. They're not just the same. There are some that seem really, really smart, and there are some that not so much. They don't seem smart at all, which has probably got to do with where they live. If you live in the mud and you come out at dawn and dusk, you probably don't need to be as actively intelligent as something that's out all day looking for its food.
Right, and do you see individual differences?
Between the octopuses?
Yeah.
Yeah, you do. They have different preferences and different dislikes, and they have kind of different habits. Some of them will like to get into the flow of, a jet of water flow. Like this means water flow somehow on the screen. They'll get into that flow and they'll kind of hang out there, and it looks really weird because they're hanging on, and some won't ever go there. So yeah, they definitely do different things.
I always find that really interesting, the differences. Like you say, between species you have these differences, but then actually within the species or within a population you have variation as well, and that's always really fascinating.
Yeah.
Have you seen much tool use by your octopuses?
Tool use.
Or playing with toys?
Yeah, well, they'll definitely manipulate stuff. So yeah, we give them lots of things. We call it enrichment. So instead of just feeding an octopus and handing it its food, you want to give it its food in a way that it's got to work for it, just like it would in the wild. Otherwise, they just sit there and all they'll do is eat food if you hand it to them. Kind of like a 6-year-old, same thing. So we do all kinds of things to enrich them, which can be as simple as live food to as complicated as putting the food in a jar and screwing the top down and making them learn how to open the jar to be able to get their food. Or I like to actually put their food inside a pirate ship, and because that's funny to have a little pirate ship with an octopus trying to pull the food out, and have little men in it that scream when they get food in. Things like that. Yeah, so they do play. Sometimes I'll stuff food in a wiffle ball, and they'll either try to pull it out slowly, or they'll just push their mouth parts against the hole in the wiffle ball, and with their arm tips, they'll push the food against the hole so they can eat. So they can figure out how to cheat. Yeah, definitely want to keep them entertained. Otherwise, they get bored, and if they get bored, they can start doing things you don't want them to do, like trying to figure out how to escape.
Right.
Now, has that happened to you before? Have you had them escape on you?
I've had them escape. When I first started keeping them, I took really seriously the idea that they were escape artists. And I used to go through all kinds of crazy things to make sure that they couldn't escape. Some of them are really prone to escape and some aren't. The answer is yes, I've had one escape. I haven't had one escape and then, to be found months later under the couch in the other room or anything like that. I've always heard them escape and been able to put them back in their tank before anything terrible has happened.
You have to constantly monitor them.
You absolutely do.
So your interest has been, you've been working with the Pacific striped octopus. Can you tell us a bit about that octopus in particular? Yeah,
the last few years, me, Roy Caldwell, and Chrissy Hufford have been working with the larger Pacific striped octopuses, which is cool, except there's a lesser Pacific striped octopus, so they have the same acronym, so it's really annoying, and it didn't really strike us that way until the paper was recently published, and we went, they're both LPSO, that's terrible. But we didn't make that up, so it's not our fault. They're a really, really cool octopus for a couple of reasons. One is their behavior, and the other is the story about why we haven't really known anything about them until recently, until we got some specimens about 3 years ago. And that was Arcadio Rognice, who's one of the other authors on the study, tried to publish about them in like 1982, but what he was saying they were doing was so strange that it didn't pass peer review. So kind of 3 years ago when we got them, and then we saw that they did everything that he said they did, it was kind of like cryptozoology come true. It's kind of like we got Bigfoot in the lab, and it turns out Bigfoot does run in big dopey lopey strides across across the field, which there's no Bigfoot and they don't do that. But this is kind of like that, if that were to happen. So that's pretty cool. Oh,
now, were you expecting to get your Bigfoot octopus when you first got them at the Academy?
No, I never expected to see this animal ever. Before we got them, they only existed in the literature as a reference and as 2 short paragraphs in notes of a different paper. And this was like the octopus that all the octo nerds would look at and go, oh, oh, that's really cool. If you even heard of it, you know, all the ones who have been around long enough said, what about this octopus? And never dreamt we could get them, because how is that going to happen? No one's heard about this thing or seen one since, you know, the late 70s, early 80s. And we were working with the lesser Pacific striped octopus, which is an insanely interesting and weird octopus in its own right. And the collector that we were working with said, hey, we collected an octopus, and it's bigger, and it looks kind of like this one. And I remember getting an email from Roy, said, this is it, this is it. And I read what it was, and it was like, I know what that is too. And we were all kind of screaming. And then we had about a week of desperately making sure that we got those octopuses. So very exciting, but no, I never thought I'd see it ever.
And how many did you get at first when you got them?
The first one we got, I'm trying to remember, I think the first one we got was just one. And then the next week we got 4 or 5 more. And my head exploded. You know, and it's I unpacked it, and it was beautiful. It was everything that we thought it would be, and it looks like the drawings. And you know, they've got stripes and they've got spots, and they're just gorgeous, and they're very cool octopus. So
but what really makes them remarkable is how they act together, right? So if you'd only ever had the one, you might never have seen some of this amazing stuff.
That's right. That's very true. And it's how they act and how they breed and how they have their babies. So people in the media are calling it a social octopus. It appears that they live in aggregations of up to like 40 octopus in the same area, which is a really weird thing for octopuses. Generally, octopuses are just loners. Even weirder, they seem to share dens, which is incredibly weird. Even weirder than that, males and females seem to share dens, which we had happen in the aquarium, which was insanely strange. You know, normally keeping octopuses alive, you keep just one because they tend to eat each other, and that's suboptimal for display and suboptimal for long-term care when they eat each other. So that's all very cool. The other thing that's super cool, besides how they mate, which is different than any other octopus, is that they have multiple clutches of eggs. So you know, every Mother's Day you get that story of the octopus gives all for its babies, and oh, the mother octopus tends the one clutch of eggs, and then they hatch, and then she dies because she's lonely now. And so call your mother, right? That's the story. And most octopuses do that. One clutch of eggs, that's their breeding strategy. Boom, 1 billion babies go out into the wild, and that's it. And even the ones without 1 billion babies, they tend to have just one clutch of eggs. This octopus, we had one that was laying for 5 or 6 months continuously. So they're just continuously laying eggs, mating every day, and so we have, we don't really know how long it takes the eggs to develop because they were just laying them concurrently. So every day more eggs would show up. So we kind of know when the first ones were laid, but we have no idea if those are fertile or if we were keeping them colder and that had anything to do with how long they took to develop. So we'd like to get out in the field and see what they're doing there, and we'd like to get more so we can then run some more studies and see what they're doing. And the coolest thing, I'm going to interrupt myself. Oh, that's a cool thing on the video right now. That's a slow bounce pose where they kind of walk around and they kind of, it's a weird behavior. They kind of look like a dead leaf, but we don't really know what they're doing or why they're sneaking around. That's my favorite thing that they do. But what seems to have captured most people's attention is the way they mate, which is beak to beak. So an octopus, if this is a pentapus, so just imagine there's 3 more arms here because I want to do it. Right in the middle is their mouth, right? Cephalopod, it means all their limbs are in a ring around the mouth part. So right in there is the buccal mass, which has got the radula and then a parrot-like beak. And it's really parrot-like. It's like a bird's beak. It's crazy. Most octopuses would mate, let me see here. Here's the one, and then the male's over here, and they check each other out. They don't want to get eaten. And then the male jumps on the female back on top of her mantle and snakes one arm into her mantle cavity and then drops a spermatophore, which is nice and safe because then he can run away and not get eaten. And then you have one octopus left. What these guys do is they mate beak to beak. So it's all out there. If they're going to eat each other, they certainly can. They line up their arms with each other. They line up their suckers, and they mate just like that. And they do it a couple of times a day sometimes. Here's the video of it. That was a crazy night in my living room. Actually on the dining room table. But that sounds weirder. And they mate like that, and they pass the spermatophore, and the ones that were cohabitating would actually sometimes eat in that position. So they're yeah, very insane, very strange octopus.
So of course some people have said they're kissing.
They don't have lips, so no. But the kissing octopus is pretty cute. It's a cute name, but we didn't like it.
And you guys still don't have a genus species for this guy, right?
No, it's undescribed. Although as we had hoped, it's now seems to be being worked on more than it was before.
Do you have something in mind?
I don't think it's up to me. Well, the main thing we wanted to do was get Arcadio rodnice involved. So what we thought originally would be Octopus rodnice, if indeed they stay in that genus.
Yes.
Secret information.
Oh,
so there's a question. Oh, go ahead.
Yeah, go. But now it's going to be something else because he's an author, so you can't name it after an author.
An author.
Yeah.
A question from the chat room. Ed from Connecticut is saying, in a group, do all the females breed multiple times, or is there some kind of hierarchy?
No idea. I don't know the answer to that question. There might be a hierarchy. I've never had that question before, so that's awesome. There was a couple of things that happened when I put a trio together, 2 males and a female. They lived fine, but one of the females who had been on eggs then ate her eggs, and then I switched those out, and then one female was laying eggs, but another female never would. So there could be a hierarchy. I don't know. We absolutely want to get into the field to see what's going on. You know, one of the things that has become pretty clear with this paper is how important both in aquarium observations are and how important out in the field observations are, and to have both of them because they inform each other so much, especially with behavioral stuff. So we really want to get out there and see what they're doing.
Yeah, right. Well, it's in the aquarium or aquaria, like you said, you can switch different individuals in and out of tanks and see how the changes in the group dynamic, how group dynamics change as a result.
Absolutely.
And so you can actually ask very specific questions. Out in the field, you're going to be limited to how many individuals you can find and when and where you can find them, and then pretty just observational studies, right? Sure.
But also you got to know, at least with these guys for sure, is we were limited with the numbers of animals we had in the lab as well. Yeah. So that's even scarier, because what we'd really like to see is what would happen if you put 2 males together. We think they would probably fight pretty incredibly, but that experiment's got to be done at some point. But we never would do that because we didn't have enough there was a whole lot of things we would have wanted to do, but we wouldn't do because it's too risky. And so we'll see. And then, and trying to breed them and close the life cycle, which we weren't able to do, but we were able to get babies to eat, which is a big step. That would be the goal, and then we could study them at our leisure rather than, they have short lifespans, so that's the other sad and crazy thing about them. A year to 2 years, and then that's it.
Oh, so that's really short.
That's really short. And this octopus is in cooler water, about 19 degrees C, which is what, like 69, 70 Fahrenheit. So generally, the cooler water they're in, the longer they live. So we figured this one's about 2 years, and we seem to be right on with that. But your tropical octopuses, a year, 10 months to a year, and that's it.
Wow, I never thought it would be that fast of a life cycle.
So fast.
Yeah, so I wanted to ask you, since we have the octopus expert, that last summer we reported on a story where they found an octopus off of the coast of Monterey that had been taking care of her eggs for 4.5 years.
Yeah.
In this really cold water. And so is this something else that is unusual, do you think, or do you think that it's just there's so much stuff going on in the world of octopuses that we don't know about yet, that this could just be totally normal octopus behavior?
I think it could be totally normal for the cooler water octopuses for sure. While it seems like we know a lot about octopuses, there's so much we don't know. It's just massively understudied, just massively. All the cephalopods are, and we should fix that.
Well, we definitely need to get into the oceans more and learn much more about the animals and the interactions that take place under the sea. Yeah.
Under the sea, at all levels of the sea, because the sea is deep, and different things happen at different deepnesses. Deepnesses, you like that? Deepnesses?
Deep. That was pretty deep.
I do want to say I'm so happy that we got to talk about this on the show, because I heard you talk about this larger Pacific striped octopus at TonmoCon a couple of years ago at the California Academy of Sciences, and you talked about this beak-to-beak behavior, and I could not believe it. And I was combing the journals, waiting for it to pop up, and when it popped up, I got so excited because
she did really get quite excited.
Thank you. That's great.
So it's awesome. I'm so happy we got to bring you on the show and talk all about it. Loyal listeners will know that I get very interested in cephalopods and also in invertebrate sex. So this is like the perfect amalgamation of the two.
Perfect.
It is.
Yeah.
So,
thank you.
Is there are you looking at any other specific behaviors with this particular octopus moving forward, or what are you doing?
Hunting. What are we doing? We have one specimen now that was caught this year, and there's stuff we want to look at. Their feeding behavior is another thing, and they clean their houses. They don't really have houses, but they do clean up. They're reaching over to tap the shrimp on their butt to make the shrimp run towards the octopus so they catch it easier. Is pretty neat. So we want to look at that. And all kinds of stuff. The mating behavior, we still want to see what's really going on. It's just kind of everything. We just really have the first course. And the next courses, I think, will show us a whole lot of stuff that I think everyone's going to find fascinating. Or at least me, and that's all I care about.
Exactly. It
maybe me and Blair will be the only ones.
No, it sounds like you've just got the perfect job for your interests. Just perfect.
I think my job's pretty perfect. It's kind of hard to complain about my job, except for having to fill out TPS reports. But besides that, it's a great job. Earlier this year, I was out in Hawaii collecting the day octopuses, big octopuses, which is like your octopus's octopus. When you think of octopus, that's the one. And I'd never worked with that before. I never collected it before. And I'm out in the water with the team, and we're swimming around looking for octopuses. And, it's like, how did this happen? How is this my job?
Not a bad day.
Not a bad day. And then we caught them all the ones we were supposed to catch, and they shipped well. And it was great. So yeah.
Always consider where your field work will be before you really dedicate yourself. I mean, you may be fascinated by Arctic archaeology, but where you're spending your summers is going to be a whole lot different than if you were going to be studying some sort of tropical archaeology.
For sure. And then working at the aquarium and the academy is pretty fantastic as well. It's a great facility with great support and great staff, and we're getting to actually interact with the public very often because all the exhibits are on display. If there was a job that's better than juggling or glass blowing or improv, this seems to be it.
Yeah, the only thing I'd say that I'd have to complain about with that job is there's no paper towels at the academy.
There are paper towels for me.
Yeah. Behind the scenes.
Just not for you.
Yeah.
The public restroom with the blowy thing that's always got a few too many examples of who was there before I can't use those.
There's always some kid there, so you can just use their shirt.
Just air dry it.
Let me ask, which octopus species are on exhibit right now at the Academy?
Right now, we've got the day octopus, Octopus cyanea, and what else do we have? The giant Pacific octopus, the giant Pacific octopus, which is very different than the lesser, the striped Pacific octopus. This is the one that gets huge. That's on display, and I think at the moment that's all we've got on display. Like I said, mostly short lifespans, so and hard to breed, so it becomes difficult to keep them on display all the time. Now we do have other cephalopods on display. We've got nautilus on display, which are laying eggs, which is pretty fantastic, and we've got two species of cuttle. We've got the dwarf cuttle, Sepia bendensis, which are on display and they're breeding, and the flamboyant cuttlefish, Metasepia, which is also on display and breeding. So we're good for cephs. I think we've got a nice ceph collection right now.
That's my favorite cephalopod, the flamboyant cuttlefish.
I've got a question. I've heard it said, I've seen it told, that octopuses can recognize people, that they have a facial recognition of people, that they will sometimes treat one researcher different than another. Have you seen any examples of this?
Yeah, that's very Lamarckian. So I'm just making fun of you. They very quickly learn who comes with the shrimp. So they know that, and if in the home lab here, the secret home lab, I'll go in, and any of the cephalopods will come up. My wife says, "Shrimp come with people. People come with shrimp." And all the cephs, the cuttles will at least rise up. This is what a cuttlefish looks like with your hand, by the way. And they'll swim right up to the glass, and they'll look at you and follow you as you move across the room, or at least me. But somebody else comes in the room, they're not as excited. So
wow.
So that's definitely. The paper is pretty compelling about that, how they were able to show that they recognize different people. So, again, do all cephalopods do that? Who knows? But there are definitely some of them that appear to do that. Definitely some that appear.
That's
way to double down on
absolutely non-committal.
Well, we have, because we've reported that story before in the show, and then it just seems really fascinating to me.
Yeah, well, they've got good eyesight, so.
That's what I was going to say. Their eyesight is on par with people, right?
Yeah, except they apparently they don't see color.
Yeah.
Like apparently me and Blair. So, but
so many things in common here.
I know, it's crazy. But their eyesight has to be good because they need to eat, and so they need to hunt. But they're also delicious, and everything wants to eat them. So they have to be able to get away from those critters that want to eat them.
Awesome. Well, we want to let you get away from us if you must go.
Nice segue.
No, I'm every week, and we're going to have the same discussion now.
Oh dear. It's slightly repetitive. Thank you so much for joining us. Can people follow you on Twitter or on YouTube, or are there places where you share your cephalopodic news?
Sure, I'm at Twitter at Cephhead. I'm not very good at Twitter, so it's kind of there. I'm on Facebook at Richard Ross, and I've got a website called richross.org where I put stuff up. So there's blogs and scrapbooking things, and all the work I've been doing I kind of keep there, so it's all in one place.
Wonderful.
And then at the Academy, of course.
Yes, and California Academy of Sciences at Cal Academy. They are open October 8th, so you will be able to celebrate World Octopus Day.
Absolutely.
Yes.
And you should. You should.
How are you going to celebrate World Octopus Day?
I am going to pick up some shrimp in the morning so I can feed cephalopods. And then I guess at 10 o'clock I'm doing a Reddit Ask Me Anything for a couple hours.
Oh, how fun!
Which I'm kind of pinching myself over. And then I get to do the rest of the work I'm supposed to do on my normal day. So taking care of animals, making sure they look good.
Fantastic. Well, thank you again. We really appreciated you joining us here tonight. It was informative and fun, and I hope that you have a wonderful World Octopus Day.
Thank you. My pleasure. Thanks for having me. I really had a good time.
You're welcome. All right, everybody, this is This Week in Science, and we do have a few little tiny science-y tidbits to end the show on. So, Blair, you want to tell us about the new panda?
Yes, it's called Cactus.
Huh?
Will you explain this? I don't understand.
No, I won't explain it at all.
Okay.
That's all you need to know now. There'll be a big, marvelous, amazing announcement later on.
According to the first comprehensive global assessment of species by IUCN and partners, published today in the journal Nature Plants, cactus are amongst the most threatened group of things in the world.
Really? Does that
Yes.
have to do with climate change?
It has to do with trafficking.
Oh.
Yeah, so more than half of the world's almost 1,500 cactus species are used by people, and 47% of the threatened species are threatened due to illegal trade in horticulture and private collections, as well as unsustainable harvesting. So cacti are among the most threatened taxonomic groups ever recorded. And this is a problem because cacti are kind of important. So they live in places where it's kind of hard to live, no matter what kind of living thing you are. And so a lot of animals and other plants depend on cactus for certain functions in the environment. A lot of animals depend on it as a source of food, a source of water, a source of shelter. All those things, when cactus start to disappear, those animals are in big, big trouble. And so it's very interesting that these cacti are this extremely threatened group. And I never knew that cactus had so many pressures coming from illegal trade, for example.
idea.
A lot of it also comes from clearing cactus habitat for things like shrimp aquaculture.
Interesting.
Yeah. So there's all sorts of weird impacts on cactus I had never known about. You can read the full report at IUCN. But essentially, cactus, unsung heroes of the threatened species.
Which is not a good thing.
Yeah. So
So
I urge everyone to find out. I don't even know how you would do this besides going to the IUCN website. And so people are working on getting all these cactus species listed with a lot of information. But so before you buy cactus for your home, for decoration, for your office desk, or before you hire a company to do landscaping for you, you should make sure that all of the plants are sustainably harvested or grown.
Thank you. I've got some news about ravens. Ravens are especially particular species are known to be tool users and incredibly smart. Now, a new study at the University of Vienna suggests that ravens also cooperate. So they gave ravens an experimental setup where there was a string and cheese was on the string. The string was through a tube, and the ravens both had to pick up, two ravens in a cage, each had to pick up the end of the string to pull it through the tube so that it didn't get dropped. If the string got dropped by somebody, then the cheese was lost for everyone. And I just suddenly went to it, the cheese stands alone in my head. But anyway, these ravens, it turns out they cooperate. But sometimes ravens will just, an individual raven will just be like, that's my cheese, and will steal the other raven's cheese in addition to their own cheese. After only a couple of experiences with that, a raven who has had their cheese stolen learns and remembers not to trust that other individual and stops working and cooperating with individuals that steal cheese.
Oh.
So ravens cooperate,
but is willing to
with
cooperate
everyone.
again. But is willing to cooperate again with a raven that didn't steal, or is
Yes,
just jaded against cooperation altogether?
If another
individual
raven,
specific.
it's individual specific. So if there's another raven
That's
that
cool.
does cooperate, those ravens will continue to work together.
That's cool.
That's a multi-level smartness right there.
Yeah, and researchers are saying that this kind of level of social record keeping has only really been shown in humans and chimpanzees, and so in birds, this is something very new. Now, in world robot domination, our robots, our robotic cars, our robotic everythings, they're going to know what we're thinking.
Uh-oh.
Yeah, a researcher at the University of Illinois at Chicago, graduate student, has published a study in PLOS One about an algorithm that he developed by watching people. He let people like reach for an object on a table and then push their hand out of the way and recorded what happened. So what happens to your hand when it is interrupted or disturbed from its path? Based on that information, was able to come up with an algorithm that could help cars determine where you meant to go before you had to swerve to avoid an obstacle. To allow some kind of a vehicle or other robotic device to know what your intentions were, so that at the end of a movement, because you've been pushed out of a certain vector, that the car will help you return to where you want it to be as opposed to pointed at a tree now. So the idea is that because our brains take a while to figure out that the hand that is outstretched has been disturbed, there is about a 1/10 of a second to a 25th of a second delay, a 1/10 of a second delay from that information of the hand being pushed or the steering wheel being turned, for it to get to the brain to figure out what happened, and then the brain to send new instructions down to the limbs. And so because of that, there's a weirdness that happens in our limbs, and that they're able to analyze and now are developing algorithms. And so algorithms for predicting how we wanted to move, even though we didn't.
Fascinating.
Yeah, artificial intelligence to make us safer. That's the idea. Another idea is for prosthetic devices. So people who have palsy, and I think this is actually the most interesting part. For people who have palsy, if your hand is shaking and you want to move and pick up a cup or a pen or some object, but your hand is shaking too much to get it there, if you have a prosthetic device that knows where your arm wants to go, and it can assist you.
Great.
And that also seems to be a problem of figuring out ways to dampen signal that's being sent, because in the palsy, the signal is being sent to the muscles to activate and to activate. There needs to be just a dampening of that, it seems. In the cars, they're already beginning to keep lanes for you. They have forward-facing cameras now that can watch the lanes over a certain speed, and if you're drifting out without having a turn signal on, it can gently nudge the car to the middle of the lane again. This is a feature that's available on a lot of new cars coming out this year, as well as crash mitigation is a new feature that's pretty cool.
Yeah.
Where they will, if it senses an impact is going to happen, it's like this quick stage. There's a light and sound alert. There's soft braking. Then there's hard braking. I had a customer who had one of these cars, had this brake mitigation, and got into an accident, hit a deer. Didn't stop the car from hitting the deer, but this isn't something ahead. This is something that jumped across the traffic, right? But her report was she felt the car braking before she even saw the deer. So instead of hitting it at 60 miles an hour, hit it at maybe 45. It reduced the impact of the accident, which then can reduce damage to the car, the deer, everyone involved. So we're getting to those the stage where not necessarily that the cars are going to drive themselves tomorrow, but that it's making it a lot more sort of idiot-proof, the driving experience. There's already also things that manage the braking and the speed if you make a turn and you're going too fast to make that turn, where you would normally lose traction with the road and go sideways. These vehicles can now apply some braking, downshift themselves so that where you point the car, it'll go. So there's been a lot of this safety technology from the driver end that's positive.
And I think that the part of this that's really interesting is that because of our consciousness and our awareness, we're slow. Like our awareness of the world around us actually slows us down.
Right.
And so a computer in a car does not have to be aware. It just needs to sense. Mm-hmm. And so it's going to be faster than we would be at responding. Yeah. And then my final story has to do with sex. And this is a really fascinating sex story. Published in 2 papers, Fertility and Sterility, and the journal Physiology and Behavior, researchers from Indiana University tracked women who were having sex and women who were abstinent. And they looked at changes in the immune system. They found that very specifically, sexual activity triggered changes in immune system proteins, specific immune system proteins, and in antibodies. So women who have sex, having sex changes how the immune system functions, the female immune system. And the question is, there are 2 factors to this. The immune system has to keep a woman fit and healthy, but it also has to not attack a potential baby. So what they found is that there are certain T cells, type 1 helper T cells assist in defending the body against threats from the outside, and type 2 helper T cells help the body accept things related to pregnancy so that they don't get mistaken as attackers from the outside. And so they found women having sex had significantly higher levels of these type 2 T cells during the luteal phase of the menstrual cycle, which is when the uterine lining is thickening and preparing to host an egg. So it's in that phase when a woman is about to get pregnant or could get potentially pregnant, these type 2 helper cells are going, yeah, okay, let's get ready for baby. Let's not attack stuff in the pregnant area. Yeah. And then they also found these women had higher levels of type 1 T cells during the follicular phase, which is when the ovaries, the follicles in the ovaries are maturing. So they're not quite to the point of releasing an egg yet. There's no egg in the fallopian tubes. There's no egg that could be fertilized at that point. There's just a maturation phase, and so the type 1 is higher just to protect the woman in general. And then additionally, there's higher levels of immunoglobulin G during the luteal phase and immunoglobulin A during the follicular phase. And so none of these changes were found in the abstinent women. And so the big take-home message to all this is that having sex could be part of the reason that some women get pregnant kind of outside of the normal period of when you're supposed to be fertile. Sometimes, that there could be that having sex could actually be triggering immune system changes and other changes within the female body that make it more receptive to pregnancy. And so the idea of, hey, if you want to get pregnant, you should have a lot of sex, you should.
That's one way of doing it. That's just one way of doing it.
Seems like the most obvious way of doing it.
Right, yeah. But I mean, some people would just have sex during the time of the month when there's, it's like, okay, I don't have to have sex all the time. We could just have sex during this one little window. That'll be fine. But it might not be. Right. And this might be why. Wow. So, but this is really like one of the first times that that sex has been shown to change the human female and fertility. Interesting. Any other stories, you guys? Nope. All right, we're all out of stories, all out of fun, but there's more science out there, and I would like to take this moment to thank our Patreon sponsors for supporting us. Thank you, Kevin Donald Wesley Ballard, Ed Dyer, John Ratnaswamy, Steve DeBell, Rudy Garcia, Gerald Sorrells, Greg Guthman, Dave Neighbor, Jason Dozier, Matthew Litwin, Eric Knapp, Jason Roberts, Patrick Cohn, Shane and Tara Ginsburg, Richard Onimus, Byron Lee, E.O., Bob Calder, Jared Lysette, Ulysses Adkins, Brian Condren, Jake Jones, Trainer 84, Kati, Edvardus Rimkus, Brian Hedrick, Cassie Lester, Sarah Chavez, Stephen Stroyak, Layla, Marshall Clark, Charlene Davidson Henry, Don Kamarechka, Larry Garcia, Randy Mazuka, Tony Steele, and Dave Friedel. Daryl Lambert, Kevin Parachan, Robert Aston, Stephen B., Dave Wilkinson, Stephen Mashinsky, Phil Clark, Rodney Lewis, Braxton Howard, Phil Nadeau, Rick Ramos, Salgood Sam, Matt Sutter, Emma Grenier, Philip Shane, James Dobson, Steve Goodwin, Kurt Larson, Stefan Insam, Michael George, Craig Landon, Russell Jensen, Mountain Sloth, Jim Drapeau, Tara Payne, Alex Wilson, John H. Maloney, Jason Olds, James, Noah Wiles, Paul West, Alec Doty, Illuminlama, Joe Wheeler, Dougal Campbell, TMRO, Miko Pakala, Craig Porter, Adam Mishkan, Aaron Luthen, Marjorie, Paul Stanton, Scott Lajewski, David Simmerly, Tyler Harrison, Ben Rothig, Columbo Ahmed, and Gary Swinsberg. Thank you so much for all your support on Patreon. And if you're interested in supporting us, you can find information at patreon.com/thisweekinscience. Also, remember that you can help us out just by telling your friends about TWIS. Thanks again to Dr. Richard Ross for joining us today to talk about our favorite underwater invertebrates. And don't forget that this week is Cephalopod Week. Hug a cephalopod today? Or not. On next week's show, once again, we're going to be broadcasting live online at 8 p.m. Pacific Time on twist.org/live. You can watch live and you can join our chat room, but don't worry if you can't make it. You can also find our past episodes at twist.org/YouTube or just go to twist.org.
Yes, thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have one of those mobile-type devices, you can look for TWIS, the number 4 droid, in the Android marketplace, or simply This Week in Science in anything Apple marketplacey.
For more information on anything you've heard here today, show notes will be available on our website. That's www.twist.org, where you can also make comments and start conversations with the hosts and other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at Dr. Kiki, at the Science, at Actionfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you have learned anything from today's show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world, so I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice, show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand, and all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Aye, 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 aye. 'cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
Very nice.
That octopus puppet is
That is awesome.
awesome. What you got there, puppet?
It's a gift. I have to get rid of him soon. Not for me.
Uh-huh.
So I thought I'd have fun with him before I gave him away.
That is really cool.
That is
Do you
so
already
awesome.
have one?
No. I just have my stuffed animals. I have a stuffed animal.
You should have gotten 2 of them before you gifted.
I have both of these, so I feel like I kind of have the cephalopod thing covered.
Yeah, but they don't like the other one. Okay. Aw. Is that what you cuddle up with at night?
Yes.
Is it a hat or is it a cephalopod?
It's both. I have my squid.
It's a cephala hat. Cephalacap?
Head hat. That's what that means. I like how Rich Ross's Twitter handle is head.
Head. Cephhead.
Cephhead means head.
Cephhead. Head. Oh, geek jokery. It's hilarious. Rich said he had to jam. Thanks so much. That was such a good time.
He was awesome.
Yeah, he was great.
He is super fun. I filmed stuff at the Cabrillo Aquarium in San Pedro.
Yeah, you said your parents were your production crew.
Yes.
Yeah.
So we'll see how it all turns out. So I have video, which I don't know how good the quality will be. We'll see. And then I have audio that I did via a rig with my iPhone. So if I can learn how to use iMovie.
Yeah, if you want to try doing it, you can do it.
Yeah.
If you want to have a little project and do it yourself, that's awesome. Or you can send it up. You can send it to me. We'll use Dropbox or...
Sure. Let's let you have your trip.
I will have a trip. I'm going to take a trip.
So you don't have to worry about it during that time, and I will fiddle with it and probably get frustrated and then send it to you after your trip.
Oh, I have mail for Justin.
Oh, nice. What do you have? Is it a handy horse?
I have a handy horse for Blair and a number of magnetic finger puppets. Some letter from the Princeton University Press.
Ooh.
The Flicker Men, which is a novel. I started reading it. It seems interesting.
Nice.
But I could just give it to Blair if Blair wants some sci-fi reading.
Oh my God! I have a stack, like 10 feet tall of books that I'm supposed to be reading.
And the letter from Princeton's probably just an alumni letter. They're just fundraising.
Oh,
Yeah.
I'm so sure.
What else do we have? There's a book.
Dave with the office.
A book called Follow Your Gut.
Ooh, I bet Justin likes
Impact
that
of Tiny Microbes.
I like that idea.
It looks a cute little book. And then the Book of Science.
Oh.
I don't know. I could keep these or I could bring them to you, but you're not going to be there.
Yeah, just give them to Blair.
I could just give them to Blair.
Make sure they get to Justin.
She reads.
Have you guys No, I don't. Have you guys started reading those sci-fi books that we're supposed to be in?
No.
I started, but I didn't get very far.
I haven't been reading anything. I haven't been writing anything either. What have I been doing with my time? I haven't created anything like creative or like really consumed a whole lot of creative things either. I've just sort of been...
I have a lot of books.
Kids and work and...
Movement on the calendar front.
Happening to me.
Yeah, how's that going? What's going on?
So all of the graphics are done. Now I just need to find a place to print them.
Oh, cool. Okay.
So I'm going to do a little bit more fact-finding. Our lovely TWIS listener / minion, Patrick, has been helping me. And so he gave me his last kind of 2 cents about that and did the designing of, like, framing my pieces of art. So it looks really awesome. And so I just have to get a couple price points, and then I will chat with you, and then we will put them available for order.
There's a
cool.
For this.
What?
Yes. Yeah, it needs to be. This probably needs to be done within the next month, by the end of October.
I was thinking if we had it available to purchase, within the next couple weeks was what I was hoping for.
Just so we have time to advertise, so people can start collecting them to give as Christmas presents.
Yeah, exactly.
Once we, what we should think about is once we figure out how much, who we're going to go with and how much it's going to cost to print each one, then we can figure out the price point for the calendar.
Right, and then we have to decide if we're going to just order a lump sum of them.
Yeah. But as soon as we know how much it's going to cost, once we figure that out, we can set up a pre-order button, and so we can take orders.
Clever. Oh, clever.
And then then we get our printing fulfilled. We can start putting it out there for sale, even though we don't have calendars in hand to mail.
Yeah.
But then we can just start. We can start collecting orders and see.
You like that better than a print on demand?
Print on demand is probably 2 to 3 times as much.
Right.
And we won't get we won't it'll be more expensive for the listeners. Yeah. Which I'd rather do it differently. Yeah.
All right, kids, I gotta run. I gotta go.
I have one question for you, Justin. Could you go by the station at some point in the next week to check for a book? I am scheduling Paul Ehrlich for a guest on the 21st, maybe. And he's got a new book called The Annihilation of Nature. Human extinction of birds and mammals. And I don't know if they have sent us a copy already, but if they did, it would be at the station. So,
is it released already?
Yes.
The book? Okay. Yeah. So it might be at my local library.
Yes, it could be at the local library also.
Okay, great. I'll check.
Yeah. So, it looks looks like it'll be beautiful. It looks like it'll be a beautiful book. So,
not such a beautiful topic.
Not such a beautiful topic, but it would be nice for at least one of us to know what the book looks like and what's inside of it if we're going to be talking to Dr. Ehrlich.
So if you email me the or text me or whatever's easiest for you, his name and the title of the book, I will try to grab it from a San Francisco library for myself.
Okay. Sounds good.
On the 21st, you say, so in 2 weeks.
In 2 weeks.
Okay.
Yes.
Okay. I can manage that.
And then you can take the library book with you And eventually I'll get the other book over to Blair, and she'll be able to take that one back to the library and be like, your barcode fell out. I don't know what happened. But it's the book. And we'll figure it out.
I don't know. Yeah. Our chat room is very quiet as well. I think everybody must have overdosed on The Martian this weekend.
I haven't seen it yet.
I haven't seen it either, but it's playing like right down the street from my house.
Hey, the three of us should go see it when you're in town.
That would be awesome. I'd love it.
How fun would that be, the three of us going to a science film?
Oh, we could totally Mystery Science Theater it. But it would be the TWIS treatment.
That wouldn't work in space.
That wouldn't work. Yeah.
Nice. Okay.
Yeah.
Well, be in touch with your trip. Let me know what your free time is like.
Okay.
I'd love to at least meet you for a coffee or a beer, if nothing else.
Yeah, that would be great. I would love that. Yes. I think
it would be terrific.
Terrific. Probably Monday morning would probably be or Sunday evening. Sunday.
Ooh, I could do both of those.
Okay. Yes, either Sunday afternoon, evening, Monday morning. Either one of those are kind of open in our schedule at the moment.
Nice. Yeah, that would be awesome.
Yeah. Yeah, Gord, the book is amazing. I haven't seen the movie. I've heard the movie is amazing also. We'll see. We will science it. We will science it.
Science the feces out of it.
The elf. Mm-hmm.
I really gotta go. I gotta go.
Okay.
Good night, everybody. I will see you next week. Great show tonight, as always.
Thanks. Yeah, it was a good one.
Yeah, it was fun.
It was really fun.
Cepha-fun.
Cepha-fun? Does that mean that it was heady fun? I don't know.
It's all in your ceph.
It's all in your cephalopod. Headfoot. All right, you guys. Thank you so much for joining. Oh, read the book. Read the book. But I don't know. See the movie. It's available. Flying Out. I don't know. I haven't seen the movie. I've read the book. I haven't seen the movie. Yeah. Okay, we're gonna hang up because I need to pack because I'm supposed to leave early tomorrow morning. That's what's going on. And it's time for sleeping because people need rest. Look at this. It's not even 10 o'clock yet. This is so weird.
God. This is like a record.
I know. We started on time. We had a record length to our show.
Despite a guest.
And probably because of the guest, I kept pushing things forward.
Yeah, it's good.
And chatting afterwards. There we go. All right, you guys, have a wonderful, wonderful science-filled week. I hope you enjoy Cephalopod Week and Octopus Day. Hug an octopus tomorrow.
These are my tentacles.
Or if you're like Blair, find an octopus and put it on your head. Thank you for joining us in the chat room. Thanks for watching the show, and we do hope that we'll see you again next week. You guys make this show so much more fun. Love having you guys here. All right. Take care, everybody. Bye. Stopping broadcast.